My name is Krista Hines, and I am part of the University of La Crosse, Wisconsin collaboration of Riverine Ecology Research Group for the summer of 2012. Migrating birds fly right over Pool 7 and Pool 8 of the Upper Mississippi River, which is part of the major flyway. The birds primarily involved are lesser scaup and coot. There are tens of thousands of deaths per year because they stop to feed on snails and, more importantly, Bithynia tentaculata, or better known as faucet snails. The snails could potentially be infected with parasites such as Cyathocotyle bushiensis, Sphaeridiatrema pseudoglobulus, and Sphaeridiatrema globulus. The parasite's life cycle requires them to use snails for 2 out of the 3 hosts. The first host is the faucet snail, which is an invasive species that originated from Europe. Once the parasites shed from these snails, they need to find another snail in order to be passed on to the birds. The snails we have found can either be Bithynia or a few native species such as Physa gyrinea in particular. Once the bird has eaten the infected snail, the parasite is released and lies in the digestive tract, which is what sickens the bird and eventually kills it. So what we're doing is using a game theoretical approach to find insight as to how the parasite manipulates the behavior of the snail it has infected. We're looking at the second stage of the cycle, where the parasite is transmitted after the bird's consumption of the infected snail. We designed our experiment to have multiple control and infected cultured snails and randomize which snail will be chosen for the trial. We are taping 1 snail per 9x13 tray for about 3 hours. There are 2 different time slots. One is in the morning, which runs at around 9:15 a.m., and the other is in the afternoon and starts around 1 p.m. Each snail has a chance to be exposed to both time periods by randomizing with duplicates. We used a Sony video camera and a Nikon D300 to take the pictures. The game theory approach applied to this biological situation is, in short, the snail's point of view versus the parasite's point of view. We're creating mathematical models of the snail and parasite's fitness individually. Fitness functions essentially show an organism's capability of survival. We look at choices the snail and parasite each make in regards to locomotion. Some examples while thinking about these setups as uninfected versus infected snail or snail versus parasite are: how far does the snail move, literally measuring distance? How visible does the snail want to be, where the snail is spending most of its time? How fast is the snail moving, measuring speed, acceleration, and velocity? What track or pattern of movement do they choose, measuring angle of turns? The goal is to see if these answers are amplified with more or less parasites. The ultimate goals of our research will include building and finding correct fitness functions relating to the relationships between the snails and parasites. The snail's goal is survival, which encompasses not being eaten by the bird, and the parasite's goal is survival up until it's time to be transmitted, and when that time comes, it hopes the snail will be consumed by the bird. We'll be collecting the images by using the software Stop Motion Pro and transferring them to another program to be analyzed. That software is Image Pro Plus 7.0, which allows us to use 2D object tracking that can determine distance from the origin, which can be manually set on the screen, a cumulative distance and distance from last point, acceleration, velocity, x and y coordinates separately, and many more things, but that is what we're most interested in that we can analyze and find statistics for very easily. Image Pro puts out an image that allows us to see the track on the snail in one smooth motion. We'll be taking the data that we get from Image Pro and analyzing it with multiple statistical tests, one of which being a repeated measures MANOVA. Our overall goals are to find potential explanations for these proposed questions and how, in general, does the parasite change and or manipulate the snail's behavior. In my next podcast, I will be working through the Image Pro software to show specifically how it enabled us to track our snails.